Review what I'm going to talk about. I'm going to talk about 3D cell culture, more specifically magnetic 3D cell culture. So I'm going to give you a brief history where the technology came from, give you some results that we have done with biotech. We have done a lot of work with biotech, and then I'm going to show some fairly new results that are related to a lot of the themes that are from where we are now. And finally, I'm going to show you some new assays we're developing. So, see, there we go. So just quick overview where we came from. The technology came from collaboration from Rice University and MD Anderson in Houston. I was at MD Anderson. The other collaborators were Rice University. And and we were Nano 3D Bioscience before. And as of April last year, Griner acquired the technology, which is why now I'm Griner. So I've been with the technology from the very start. So I'm sure I'm preaching to the choir, but and I'm not going to give you a high-level view of why 3D cell culture. I think if you're here, we understand why 3D cell culture is important. But this is a slide, one of the first relevant piece of data we got, which you don't need to be an expert, right? So to see that these two slides here, these two columns here are different. So this is a glioblastoma cross-section that we did the immunofluorescence for encadering. So this is the staining. You have your 2D. This is in vivo. And you can clearly see that the expression pattern is different. So why are we doing 3D? Because if you compare the two, which one is 3D looks like in vivo. So that's why we're trying to use 3D cell culture to generate more in vivo-like models. But talking about 3D cell culture, we've done cell culture for 100 years, right? We know how to do it. You take cells, put in a petri dish, they go to the bottom, they stick and they grow. A lot of cells grow very well, right? Now there's this paradigm shift. We're gonna go into the 3D world. Now let's just retrofit whatever we did for 2D in 3D. But then all of a sudden you realize when you go into 3D world that we realize how valuable it is, the cell attachment to plastic or glass. All the workflow we have, as simple as changing media of your cells. The reason changing media is easy, because the cells are stuck to plastic. So you can just change your media. You can even pull it off if you want to. If you want to do immunofluorescence, all the manipulations, you do it easily because cells are stuck to plastic. So magnetic 3D cell culture is coming as a surrogate for the cell attachment. So use the magnetic field as a surrogate for cells stuck to plastic. So to make it easier to keep the workflow very similar to 2D, but working in 3D. And of course, I'll tell you how we do this. So how we do this, first, magnetic 3D cell culture, cells are non-magnetic, right? So we do have to magnetize the cells. So that's the first step. So we have a magnetic nanoparticle called nanoshuttle. We add to the cells, and the nanoshuttle is gold, iron oxide, and polylysine. It sticks electrostatically, no specific to the cell membrane, and you just add to the cells. That's it. No special media. And then you have two approaches. We have what we call the magnetic 3D cell culture by levitation, where you actually place the magnetic field on top of the cell dish. And the cells lift off the bottom and grow in 3D levitating. That's actually where we started. But this is 6 and 24 well only. And then we have the higher throughput, which we call the bioprinting. Bioprinting, we use a cell repellent surface so the cells don't stick to it. And we can use magnets on the bottom to guide the cells. We can aggregate them, we can make rings, different shapes. So those are the two approaches that we have. And I'll go into detail. So this is just a few quick results. So here is just a picture of, because we magnetize the cells, we use magnetic field, we can bring the cells together very fast. And then they start interacting, we can generate the spheroids very rapidly. This image here where you have, sorry, I'm pointing on that side, and maybe I should point it here so everybody sees where I'm pointing to. Can you guys see? Yeah, sorry. So here on this top right, actually we had Glioblastoma, GFP label with astrocytes. We culture them separately, and then we magnetically guide them together. And you can actually see the cells crawling up into the astrocytes, the cancer cells. The left side, actually, this was a work we did with biotech. We have a whole application note on this where we did a chondrocyte differentiation from beginning to end, fully automated. And this was also collaboration with Hamilton. So we did all the washing steps for the differentiation of the chondrocytes. And the only reason we could do it is because we could hold the cells magnetically on the bottom. And so this was fully automated. And on the bottom right here, and I'll share a few more slides on this study. This is an adiposphere where we co-culture pre-adipocyte stem cells with endothelial cells. And I like to show this slide for a few different reasons. One, this was the cover of Tissue Engineering. It's a beautiful image when we published this. And the reason the image came out beautifully is because we stained the cells whole mount. What does that mean? We just fix the cells, put it on a magnet, and we could do the immunofluorescence just like you do in monolayer culture. So we didn't have to section, or because with the magnetic field, you could hold them down. So that's one of the practical advantages of the magnetic 3D cell culture. But from the biology side, we started working with, in collaboration with this group in Houston, the University of Texas. They came to us, they got our system, they came to us. We wanted to generate an adipocyte model where we can incorporate the vasculature or something that resembles a vascular process of endothelial cells. And they're interested in the relationship between obesity and cancer. So they came to me, okay, so how do we mix the two cells? It's like, you know, we can help you because we magnetize the cells, we can bring them together, and then they ask, what ratio of cells do we use? And we haven't done it. I said 50-50. So they did 50-50. It didn't work. So you get the patch, the cells come together, but you didn't get the beautiful sprouting. So we have to manipulate the culture. And we do, for us to get the green, so here, what you see here, the green is a GFP label endothelial cells, the red is pyrrolipin, which is a adipocyte marker. And for you to get this sprouting, we had to use 95% pre-adipocyte stem cells, only 5% on the endothelial cells. Otherwise, you don't get the sprouting. So my point is, if you're going to move into the 3D cell culture, more complex cultures, you're going to have to optimize. So that's a given. So, and this is the image we published. We quantified actually the angiogenesis process. The reason they wanted to go in 3D is because they also wanted the adiposphere have the in vivo morphology, which is on your right here, you have the white adipose tissue. You have these multinuclear regions with the lipid pockets. And that's very similar morphology that you get with the 3D culture, which is very different from the 2D. The 2D, the cells get these droplets. It doesn't resemble in vivo. And actually, in this work, we took a step further. Because we optimized the ratio of the cells, and we got the cells to sprout, our collaborators said, okay, we have a hypothesis here. So what about if we take exactly how the cells are, the exactly ratio that is in vivo? And for us to do that, we took the stroma of the fat pad of the mouse. We removed the adipocytes, and we magnetized the cells, and we cultured the whole stroma vascular fraction. And after we brought the cells together, we didn't do anything different. The tissue organized, which was very interesting. And this is published. So what you see here on the left, the green is CD31, which is a blood vessel marker. On the right, the green is decorin, which is stroma. So by just bringing the cells together, they organized, and they made some very organotypic like structures. And this is published. So again, it's bringing the cells together in 3D. It's a very valuable process. So now, I like to show this slide for a few reasons. We went to academia, to industry, and the best marketing, it is a good publication. So we have worked really hard in before we became Griner, we're very small, so we worked really hard in publishing good journals. And the other reason I show you this is because we use the nanoparticles. So I'm sure you're asking, do the nanoparticles do anything to the cells? And they always ask. And we've shown over and over that the concentrations used, it doesn't do anything. It's bioinert. And we have a 5-page white paper that we kind of compiled a lot of the results where we show nanoparticle with nanoparticle, without nanoparticle, data from viability, proliferation, protein expression, stem cell differentiation, inflammatory response, oxidative stress, gene expression. And actually, we have a collaboration with Scripps that I'll show some of the results in a few slides, where we did a big project. We got a NCI grant, IMEC grant with the Scripps, and before they started using our system, it's like, okay, does the nanoparticles affect drug interaction with cells? And they did all their control compounds with different cell types, with and without nanoparticles, absolutely no difference. So the nanoparticles are just basically bioinert. Um, what the system looks like, it's fairly simple. Like this is what we call the bioprint spheroid making. So you have a plate that has a bunch of pin magnets. You put a plate on top, put your cells, and since the cells are magnetized, they go to the magnet. And we'll have it over there. You guys can take a look after. And you don't need to leave the cells on the magnet. Only 15 minutes, the cells come together, and then you can take it off, and the cells do the work. With exception of one case that I'll go over in a few slides. So you leave it in 15 minutes, and the cells do the work. So this is a quick 90 seconds movie that shows you the exact workflow of the system. So it's worth going through. So the magnetic bioprint is a kit. So you have two, what we call the spheroid drive, the holding drive to change media. You have your nano shuttle. You mix your nano shuttle. You just add to the cells. You let it incubate overnight. That's it. One step. Next day you come. You wash your cells for detachment. You count. You do everything as you do with 2D monolayer culture. Nothing special. Then you're going to take your cell repellent surface. You're going to put it what we call the spheroid drive, and then you're just going to dispense your cells. See, the pin magnet is right under the middle of the well. So dispense your cells in the magnetic field, brings the cells together immediately. As soon as they hit the bottom, they come together. So 15 minutes on the drive, you come, see, they're all in the middle. And then the cells do the work. Now, you want to add a drug, you want to replenish your media before you do that. Then you put it on the other magnet, you hold the cells down, just remove your media, which is not trivial to do in 3D. And then you add your fresh media. You put it back on the spheroid drive to center the spheroids. It's important. And then you can add your compound. So the two different steps is putting either the nanoparticle and putting the cells on the magnet when you're manipulating. It's that straightforward. And here we show 396, 384, and we also have 1536. We have 6 and 24, which is very much the same workflow. So this is some results. We're using cardiomyocytes. This is, I assume some of you are working with the stem cells. This is our iPS cardiomyocytes from CDI, and we're just using our 384. We plate the cells, and you can see they start beating, and they beat can you see? They're beating. It's always nice to see. And we've done this, and they beat very consistently for several days. This is actually a poster we got from AstraZeneca in UK, in which they actually measured the electrophysiology from cardiomyocyte spheroids. And when you go to 3D cell culture, again, manipulating, doing a lot of the things we do, like electrophysiology, is no longer trivial. And here is an example where you use the magnetic field as an enabling tool to actually guide the spheroids onto the electrode. The only reason they could do this work, measuring the electrophysiology is that they could guide the cells magnetically to the electrode, and they just use the regular drive, and they measure impedance, field potential, and you can see when you have the magnetization on the magnet, you have very nice reproducible results from the beating, from the electrophysiology response of the cells. So this was very nice work that they did and shared with us. So this is another example. This is it's a co-culture we did with iPS motor neurons and smooth muscle. And this was collaboration with biotech, brain cell, which we presented at Neuroscience last year. And it was interesting because we want a nice images. We mix the two cell types. We have the neurons, which are here are GFP labeled, and the smooth muscle is red label here. And what was interesting, when we mix the neurons by themselves together, they don't do much. But then we mix them together with the smooth muscle. If you look at this image, it's just, you have your green dots and you have your red, which is the smooth muscle. But then if you investigate the cells a little further, you separate the two channels, you start looking at the pattern. This neuron seems to start to line up. So we didn't look, but it's interesting because with if you just do one cell type, it's fairly boring. But when you mix them at the right ratios, they start really communicating. You can tell that's going on. So now I'm gonna shift on some of the results we have with cancer. Of course, for many years we were very next door to MD Anderson. Yes.
When you say you're mixing the right ratios, is it identical to the ratios you would see in vivo?
No, not necessarily. Because it's always challenging. When you go to the in vitro world and the 3D world, it's about predictivity and look at the phenotype you want to see from in vivo, in vitro, but not necessarily it's the exact ratio. That's the way I view it. So we mix the right ratio to get the type of phenotype you want. Does that make sense? Yeah. So now I'm going to sort of shift gears on into cancer applications, and in these first results I like to show, and again, you emphasize the enabling tool of the technique, because this slide doesn't use adherent cells. These are leukemia cells, so they don't really form a 3D structure. So what I'm showing you here is we're using the magnetization, the magnetic field, to enable us to look at CAR T cell activity with leukemia cells. So we took NALM6 cells, right, which are leukemia cells. We magnetized the cells. And here we have the green cell is CD19 negative. The red cells is CD19 positive, okay? So we just mix them. So as you can see on the right here, actually there is red and green there. And then we had a CAR T that is targets CD19 positive. But the reason we did this, for the sake of imaging, it's much easier for us to image a circular structure that it's all in one shot than have to scan the well to find the cells. So then we had the CAR T cells at ratio 1:3 T cell to target cells, and see what happens here. The red goes away. The CAR T targets CD19 positive, so you could quantify and show that the CAR T are actually killing the cells. So, and then we started, this is in collaboration with MD Anderson as well. All this work is with MD Anderson. We are moving from leukemia cells to solid tumors. And again, using CAR T with solid tumors is tricky, and we've done some work also with the biotech. We have an application note using CAR T and breast cancer cells. In this case, it's glioblastoma. So we used the same approach. We used glioblastoma cells, U87. The red cells are PDGFR negative. The green cells are PDGFR positive. And the CAR T is supposed to target PDGFR positive cells. So we just brought the cells together magnetically. This is the first look what happens within the overnight of imaging the cells. The cells assemble and actually form a lumen. This is just the assembly of the 3D structure within the first 12 hours. So we just magnetically brought the cells together and watched them interact. Now, we did add the CAR T cells into the culture. So, and that's what I'm going to show in the next slide, that you can actually see the CAR T cells infiltrating the cells. The CAR T are not magnetized. So, and the CAR T here are blue fluorescents. So especially in this region, you're going to start seeing the infiltration of the cells start flaking over time. So can you see they're starting to, the bright spots infiltrate with time? So in this motion of the cells, which I'm going to go into it a little bit further in the talk, it's very valuable, and we're actually watching this mobility of the glioblastoma cells, how it can change when the CAR T are actually killing the cells within the culture. So, yes?
So for that example, you were just using nanoparticles to bring the glioblastomas.
We just bring the cells together, exactly.
Yeah,
We just,
onto the T cell.
that's it, not the T cells. The T cells are free floating. They're just, yes.
What's the size? What's the dimension of the spheroid or the?
So the dimension of this is spheroid, this is about 500 microns.
In all directions?
Yes, it's fairly spherical. After they grow, again, they change in size by half, if you saw. And you control the size of the spheroid by cell number. So whenever, if you, and I can show you some data too. Actually, I don't have that slide. I removed those slides. But different cell numbers will give you different size. So they all come to the center of the spheroid, which has the strongest field, which is the dot. So you control the size of the spheroid by the cell number. Of course, different cells with different cell size will give you different sizes. Yes?
For the leukemia cell lines, even though the cells usually don't form a 3D structure, if you bring them together with a magnet, they stay together.
They stay together. Yes, they don't float around. They stick together. They stay there. Because that was, it was 48 hours. So they stay together. Now, you can put it back on the magnet and bring them back together again, but we didn't do that. So they stay together. And that also depends on the type of imagery for real-time imaging. The citation 5 is very smooth. So just from experience, the IncuSite, it's not very smooth, and it clicks a lot, and the cells move a lot. Now you can put it back on the holder and bring them back to the middle, but they stay together pretty much. As you saw, that movie that you saw, that was, but those are adherent cells. That was overnight.
And is there any reason that actually you remove the magnet after 15 minutes?
Because if you have many plates, right, then you just need one magnet. You can just serially put the magnets. On this study, actually, that I'm going to show here, we actually leave on the magnet overnight, at least overnight. And this is the collaboration we had with the 15 minutes, just because you don't need it to leave. We just put it there and let the cells do the work.
Yeah, because my question is because when you use actually ultra low attachment plates, so their cells are not, they reproduce aggregates, but they're not stiff enough when you pipette them out. So I'm wondering, but if you leave the magnet over, maybe
you can. Some cells, yes. Some cells, especially the ones that are very soft. And actually, we developed these drives, the bioprinting drive, with Genentech, because there was a group that they had these cells, cancer cells, that wouldn't make a circular structure. And they want to force the cells into a circular structure because it's much easier to measure a circular structure than amorphous structure. So that's how they would just leave on the magnet to keep forcing them on that structure. But yes, you can. You don't have to, but you can. And in this case, actually, it was interesting because this was it's work of Professor Bartholomew at MD Anderson. And he did not want to use our system at first. So they optimized they did all the 3D cultures with cell lines using round bottom, the micro pattern plates. But when they went to the pressure samples, which were the PDX samples, so this is a PDX from triple negative breast cancer patients in inflammatory breast cancer. And when they took the cells from the PDX from the mouse, broke it apart, and put it on the round bottom, they would not form a cohesive structure. And that's when I was talking to him. Let's try the magnetic cell culture and see if we can make the cells stick together, because that's what we're good at. The magnetic cell culture is good at bringing cells together. So the first experiment they did, they got the system. I helped them out with the protocol to break these cells apart, and then he did as we recommend 15 minutes on the magnet. Magnetize the cells, put 15 minutes on the magnet. The cells came together. He took it off and left. 2 days later, I called him. So how did it go? He said it fell apart. It didn't work. Cells are dead. And then I told him, what happened? Did the cells die and then the tissue fell apart, or did the tissue fell apart and then the cells died? So let's leave the cells on the magnet. And actually, that made the difference. Keeping the cells, pushing them together, the reproducibility of the system of the 3D culture was much higher. So with patient samples, we do biopsy samples too, we keep them together. So this is the result. So we compared, so this is the experiment base. We took the tumor, the PDX tumor, the breast cancer, we took it out, we cut it in half, we processed as and analyzed as the traditional way. The other half, we took the tumor, broke it apart, give it a protocol, we magnetized and bring the cells together. And this is the result. So we look at ecadherin, vimentin, Cas7 pismat, and the left here. The left here is the tumor xenograft. This is the tumor xenograft, and these two are two separate 3D cultures. So if you look at the marker distribution, in morphology, it's very similar. And we did the gene expression on the paper, same thing. Pretty much, it was 95% correlation between the culture and the tumor. And we also did a dose response with the comparison with the animal and the 3D cultures correlated. Of course, you could only do 8 compounds because you cannot do a library with a xenograft. But we took a piece and we did do a full NCI library with the PDX. So that's all published now. And this is actually a needle biopsy from a prostate cancer patient. It's a bone metastatic site, of a prostate cancer patient. And we took the biopsy, we took it apart, we magnetized the cells, we brought them together, and we actually got a very nice dose dependence curve for doxo. And again, we didn't expand the cells, we just were able to really careful break it apart, the needle biopsy, and bring them together rapidly and get some nice results. And we put it back on the magnet, and you can do the immunofluorescence very nicely, which is EpChem. It is the marker of choice here. So now I'm gonna switch gears. This is the work we did with the scripts. It's a NCI grant. It's a collaboration between Cold Spring Harbor, Scripps, and us. And here was the work to validate our high-throughput system, the 1536. So it's a very dense plate. And this is just showing that the magnetized cells, when you have them on a magnet, they come together very rapidly. If they're not on a magnet, they're just dispersed in the well. And this is just a picture showing the reproducibility. Again, this is 1,536 cultures, all centered in the middle, which for imaging is very important, very reproducible in size. And a practical aspect, if you're looking at high content imaging, especially when you go to higher throughput, when you put your spheroids, make your spheroids on a flat bottom, as you mentioned, Brownian motion, they can move. And some cells move more than others. And that's what you see here. So you have always the dots are spheroids, but you can see about 1/3 of the wells you lost because they moved to the side. So we put on a magnet for 30 seconds, you can recenter all of them. And then you can image. So it's much easier that way. So now of course, we're talking about 3D. These are the results we published on the paper. And this is just showing the morphology of the cultures we worked with. So these are pancreatic cancer cells. And the actual work was using patient samples, and I'll get into the detail in a few seconds. So this is HT29, pink one. These are pancreatic cancer cells. You magnetically bring them together. In this case, they left for 4 hours here instead of 15 minutes. The reason they left for 4 hours is just the workflow of the high-throughput screening was 4 hours made sense for them. So they left on the magnet for 4 hours. The cells come together, and after 72 hours, the cell lines make beautiful spheroids. And in here, I have two nomenclatures here, AGM1 and H21, AGM1 calf and H21 calf. So AGM1 is one patient, H21 is another patient, and AGM1 calf is the cancer-associated fibroblasts from the same tumor of the AGM1. So it's a pair there, and same with the H21. What is interesting is when you look at the cancer cells, they don't really make nice cohesive spheroids. They make organoid-like structures. Even though we're bringing them together, they do what they do. They make organoids. And if you look at the calves, they make these very nice 3D cultures. So I usually mention that I'm not a biologist, I'm a chemist. So my view is cells are smart. If you bring them together in the right conditions, they do what they're supposed to do. So and this is the summary of the results. So this study that we published last year was we took, so there are two patients, right? H21 in AGM1. So you have the cancer cells, the calves, and we took the FDA library, 3,300 compounds, and tested 2D versus 3D with these cells. And on the 2D, you always got more hits than the 3D, which one way of thinking is you are, again, these are FDA approved drugs. These are not chemotherapy. So we're assuming that we're eliminating the false positives, more false positives. One can argue that you still have false positives in the 3D, but you have less. So if you're in the drug development, eliminating false positives, make things cheaper, better, that's what you want. And also what's interesting, the calves also had higher number than the cancer cells too. But was very interesting too, and this is sort of the breakdown of the previous plot. So these are the hits, and here I'm just sort of separating who is who here. So this on the x-axis you have 2D inhibition there, 2D inhibition. On the y-axis you have 3D inhibition. So every dot is a compound that we tested, okay? So if the dot is on the white, that means that it didn't work. The cells didn't inhibit the tumor. If the dot is on the pink, that means that it only killed in 2D. The green area is the overlap. It killed in 3D and 2D. And very few killed only in 3D. But it was interesting, the most active one in 3D only, it didn't do anything in 2D, is disulfiram. And disulfiram is in phase 1 clinical trial for pancreatic cancer now. So it's interesting, you know, we didn't know. So, and we're going to publish, we went further down. So I like to say that we picked the needle in the haystack in a way. But we published this is published, and we're digging into this work. And the next paper is we actually did, that was a FDA library, 3,300 compounds.
The co-culture of the fat calves that
It's coming next.
Oh.
Sorry,
Yes.
I had a question. When you said the cancer cells form organoids, but the other ones just form chemically.
Structure, an aggregate, yeah.
So how are you defining organoids versus spheroid? Is it organoid here, like multiple layers of differentiation?
That's the general approach, right? The organoids is the cells. I was in a conference just a couple weeks ago, 3 weeks ago in Holland. There's a lot of, there's organotypic, there's organoid. The traditional, the definition of organoid it comes from the stemness of the cancer cell. Yes. So, but there's the whole morphology. So the cells are polarized, you have your lumen. So did we follow if the culture, they're chronogenic, the cells that we show as organoids? We didn't look at that, but they certainly has the exact morphology of organoid, which Corpus-Spring Harbor does the organoid traditional macigel, and we got the exact same morphology. So, but good question, yes. So this slide, we just finished, we did the 3,300 compound screening. We just finished 2D versus 3D, 150,000 compound screening. And this is coming up, we're going to publish this pretty soon. And here, as you can see, if you're doing high throughput screening, the box here shows that the figures are merit of the system. So the Z prime is 0.72, which means it's excellent. Anything above 0.5 is considered excellent, right? It's the standard deviation 3 is the separation between the positive control and negative control. So it's above 0.5 is considered excellent. And the Z prime was calculated using cell Tata glow. So it's chemiluminescence, ATP, right? And out of the 150,000 compounds, we picked about 0.5%, 735 hits. And then we separated them because we do liver cells side by side. So if the compound is toxic to liver, those you further screen, eliminate those drugs too as not considered a hit. So now the co-culture. So now we took and we're doing more work. This is not published. We took the tumor, we separated the caps from the cancer cells, and now we wanted to see what happens when we bring them back together. And that's the interesting part of it's kind of it goes into the next phase of my talk, because in biology, I have a feeling that we always think of cells in a very static way. Like we take your protocol, we got a protocol for somebody, you wait for 2 weeks, and then you look at yourself. But what happens between 0 and 2 weeks? And this was interesting. So here we took the calves and labeled them red. We took the cancer cells, we labeled them green, and then we mixed the two of them together. And we took pictures at 24, 48, and 96 hours. So what is interesting is the calf makes the spheroid, the cancer cells make the organoid-like structure, but now you look at the co-culture. At 24 hours, all the cells come together. They come together out to the middle, and then after 48 hours, the cancer cells started coming out. So, and we did those dependence between and compared the co-culture, the calf, and the monoculture, and the co-culture phenotype, or IC50s, matched the cancer cells, where the calves were very sensitive. Now, again, you got to play the cell number. We're going deep into this work to try to understand. And we have a few other work groups that have done that with different cell types, and they saw similar behavior. But my point is, 24 hours and 96 hours is very different. So time is very important. It's a dynamic process. So, and even when I show this image, people say, oh, but don't you expect the cancer cells being in close contact with the stroma? And sure, but that's probably what we get at 24 hours. But the cells do come out. So, and this is a new way of looking. I'm going to go quick through this, but I do want to give you a taste of what I call time in biology. And this is an assay that we developed. If you think about the glioblastoma cells, we brought them together and they assembled in a very particular motion. And when we start seeing cells assembling that fast after we bring them together, we're like, can we use this process to measure biology? And again, we have a flat surface, which is very important. So it's really cell-cell interaction. There's no magnet when they're assembling. You just bring them together and watch what they have. So our hypothesis is, can we watch the cells and use this as a readout of biology? So this is a quick movie, and which we published in scientific reports. This is just a toxicity assay, but it doesn't have to be a toxicity assay. We've used this to fingerprint the cells. Sorry, I keep pushing the wrong button. So what you have here, it's these are 3D3 fibroblasts with their stem cells. So every dot you see here is a culture, okay? And we just took retinoic acid, vitamin A, and added as a toxicant. So high concentration, low concentration on the bottom. This is the time of the experiment. So high concentration, low concentration, this is the time of the experiment. What happens over time? In the bars that you see here is the size change of the cultures. So 3 hours, you can see when you have high dose, the cells don't move, control moves much faster. So this process of cell-cell interaction, you can actually use to quantify the toxicity on the system. But more important is, I think, is the curves that you're going to see next. The shape of these curves. So you have time versus change in size. The shape of these curves is very cell-specific. You can basically fingerprint the cell type based on the cell-cell interaction. And you can also look at drug interaction to fingerprint the drug interaction with the cells. So how do we know it's killing, right? So this is, so you have your dots at time 0, then you have 5 hours, high concentration, you can see the size change. Then we took the cells, after 72 hours, you do the live dead. Just looking at and you can clearly see it follows the live dead. And then, of course, we did cell titer glow. No, we used MTT. And here, and we can see also that the motion of the first 3 hours is equivalent of the killing after 72 hours. So this is an easy way for us to measure cell-cell interaction, drug interaction. So and in the same paper, we had the same cell type, 6 different drugs. And if you look at the shape of these curves, every drug has a different curve. So we're now trying to model these curves to see if we can infer to the drug, differentiate pathways of drugs based on these profiles. And here is a little different. So here we had the same cell type, which is 3D3 fibroblasts with different drugs, different curves. So, and now we have different cell types. In the same plate, we have A549 lung cancer, colon cancer, and hepatitis 2 liver cancer. And with doxorubicin, the blue line is the cells are not moving because it's high concentration. The cells are dead. And then you can see, but you can look at the shape of these 3 curves. They're all different for the cell type, same cell, different cell types. So now we're modeling this just using a polynomial, and we can actually use a PCA, principal component analysis, to actually separate the cell types. And there's a group at Baylor that actually using this assay to differentiate rapidly between different stem cells that are isolated. And you can really tell which cell type you have. And I'll give an example. We got PANC1 pancreatic cancer cells from 2 different groups at MD Anderson. These curves are completely different. They're not the same. So it's a simple. Now, how we did this measurement. So we take pictures of a 384 plate every 15 seconds. And the only reason we can do it, we use an iPod. It's all done with a mobile device. So the nanoparticles give you contrast, you can get a shape. So we have this, the imaging system. So you can take pictures every 15 seconds, and that's why we have, and then we can average and get very nice data. But of course, you can use other images, but this is what we used. It's very powerful, and we have some pharma companies that they're using this regularly now. So, and we can print more complex shapes. Just playing with the magnets. You can. These are just GFP labeled cells. Yes.
You're saying that with nanoparticles, you can, using the iPod, test which types of cells are dead and which are alive.
Yes. Based on this motion.
Based on
them coming together. When they come together at different rates, if they come together slowly, they're dying. I like to say, if you have people, you hold hands and say, tell them to run to the middle. If they're healthy, they run fast. If you're like drunk, they're going to run much slower. So it's sort of like that. It will vary from cell type to cell type. And this is a nice work that we just published with a collaborator in Biomaterials, where we actually took dental pulp stem cells differentiating to mini salivary glands, and we actually reimplanted it in the mouse and actually reinnervated and we showed function. This is a really nice work of this collaborator of ours. And I always showed this because last year we did an experiment at the space station. And this is Peg Whitson. She was doing this experiment right before that picture. So now if you go to space, 3D cell cultures, a lot of 3D cell culture work came from NASA. Rotating reactors, because cells in space grow in 3D. Why do you need a 3D cell culture in space? It's very hard to manipulate the cells in space. So we're just using it as an enabling tool. Changing media is not trivial. You have to exchange. So you have to carry a large amount of volume. It's expensive. Time of the astronaut to wait things to equilibrate. So we basically send magnetized cells, you put a magnet on, and then now you can just manipulate the cells much faster. So it was an enabling tool. And we tried to generate the 2D control in space by printing the cells on a membrane. I have more questions than answers. But and of course, we have lots of collaborators that got us all this done. Of course, biotech, we work with Brad Larson quite a bit. And that's all I have. I think I'll pass to Anna, and then we can answer questions.